Mycobacterium is commonly called tuberculosis, or we call it as TB. Mycobacterium, they are aerobic because they are living in the alveoli or lung region. They do not form spore and they are a non-motile rock-shaped bacteria. One of the important features of this Mycobacterium is that they are not easily to be stained by using the gram staining method. This is because they have a unique thick cell wall. This unique thick cell wall makes them resist to the staining, and other than this, they also resist decoloration by acid or alcohol. That's why they also call it as acid fast, because they do not take in the acid. That's why we call them as acid fast bacilli. For this Mycobacterium, they resist dryness and they can survive for a long period in a dry material. One of the examples like Mycobacterium tuberculosis, they are sensitive to UV and heat sensitive. If you expose them under 2 hours under the sunlight, you can kill them. However, if in the presence of sputum, which is a mucus from the respiratory tract, they can survive up to 20 to 30 hours. This is the classification of Mycobacterium. However, our focus here is only Mycobacterium tuberculosis. For the Mycobacterium classification, we also have other classification, example like the Runyon classification. This is especially for the Mycobacteria other than tuberculosis. This one you can just simply go through. The classification normally is based on the color and colony presentation when you grow this type of Mycobacterium. This is a clinically major important for this Mycobacteria. For the diseases, the Mycobacterium primarily they are causing this infection in the lung, and here our focus is only on Mycobacterium tuberculosis. First of all, let's look at the unique feature of the cell wall. For this Mycobacterium tuberculosis, the cell wall is formed by 3 major components. The first is the mycolic acid, which possesses the long chain of fatty acid. The other one is a wax D. Wax D is the major component of this fluence complete adjuvant. This wax D is the one that used to form BCG vaccine. And also we have the coat factor. This coat factor is a unique type of this glycolipid present in the cell wall. Other than the 3 major components, you also have other components that present. Example like this arabinoglycan. This links the peptidoglycan layer to the mycolic acid, and also other glycolipid and protein components. The picture here shows the cell wall structure of Mycobacterium tuberculosis. For the virulent factor, first of all, for the mycolic acid, this mycolic acid is linked from the complex link with the peptidoglycan, and it causes granuloma. What is granuloma? Granuloma simply means small area of inflammation. This mycolic acid prevents the complement deposition, which means the complement is unable to attach to the surface of Mycobacteria, and also prevent killing inside the macrophage. The coat factor is used to inhibit the migration of white blood cell. The coat factor also responsible for cause small area of inflammation, also known as granuloma. The coat factor also can be used as adjuvant in the vaccine development. While for the protein of Mycobacterium tuberculosis, it has been used in a derivative form so that it can be used in the tuberculin skin reaction test. We will explain this part at the later part when we talk about this diagnosis. Overall, so you'll notice that this high lipid component of the cell wall causes the Mycobacterium tuberculosis. They are impermeable to dye, and they are resistant to many antibiotics, resistant to killing by acid and alkali. They are resistant to osmotic lysis, and they are also resistant to killing by macrophage. For the transmission, Mycobacterium tuberculosis is normally transmitted through air droplet. For example, this person is having TB. So in the air droplet, is containing the Mycobacterium tuberculosis bacteria. So when the other person talking to this infected patient, the air droplet may be inhaled into the respiratory system. By this person will also get infected by TB. That's why for those medical staff who works in hospital in close contact with TB patient, they must always wear N95 mask. For the pathogenesis, first of all, the bacteria enter the body through inhalation, and then this bacteria enter into the alveolar macrophage. From there, they move into the lymph node. At the same time, the macrophage also will present this bacteria to the T cell. Now, in healthy person, they may develop this DTH we call delay type hypersensitivity. At the same time, they also may have this CMI we call it as cell mediated immune response. So in healthy person, what is happening is the disease will be contained, which means if you are healthy at young age, the symptom of this TB may not be shown. It will only be shown when later time, when you are in the elder age, when you have this immunocompromise, then only you'll show the disease. That's why we call them as delay type hypersensitivity. And this bacteria, it remain alive, but then they're not going to replicate. When you test with the tuberculin test, you'll give positive result. However, in those patients who have immunocompromised, for example, HIV infection or AIDS patient, they are not going to have cell-mediated immune response. Then they may progress to death. That's why most of these AIDS patients, they die because of TB. Other than this, in the recent outbreak of COVID-19, they also shows that patients with this COVID-19, they also may have subjected to this TB infection, death due to the TB. In terms of pathogenesis, as you can see, this Mycobacterium tuberculosis, when spread to the lung alveoli, from there they'll migrate into the macrophage. What is happening is there's a classic appearance under X-ray when you observe, that is the formation of granuloma. This is due to the accumulation. The macrophage in the alveoli, they try to contain the bacteria. When they try to contain the bacteria, from there, it forms a small area of inflammation, which is a granuloma. Later on, this Mycobacterium tuberculosis, they can break through and spread to other organs. That's why tuberculosis, in fact, it starts with the infection in lung. Later on, it can be spread to other organs to cause infection in other part of the organ. So the infection is characterized by the formation of granuloma and central carcission. What is carcission? It's an incomplete necrosis of tissue resulting in solid or semi-solid acellular and amorphous material. Simply, it means in the carcission, it's just a X-ray observation in which the tissue looks like firm, dry mass with cheese-like appearance. Later on, the patient may also have this Gone complex. Gone complex is the carciting granuloma. Simply means the small area of inflammation leading to incomplete tissue necrosis under X-ray observation. So, to diagnose the patient with this TB, what we can do is to conduct this tuberculin test. In the tuberculin test, first of all, we take the purified protein derivative from the bacteria. Inject 5 tuberculin units of this protein intracutaneously into the patient, and then from there we measure the induration. What is the induration? Induration is just like a small area of swollen happen in the skin, but after injecting this protein, you have to wait for 48 to 72 hours. Then only you read this diameter of the induration. From there, the positive reaction is interpreted as the 10 mm induration after 48 to 72 hours in a patient with normal immune system, which means in healthy person. However, for those patients with immunocompromised, example like HIV infection, the 5 mm of induration is identified as positive result. So when the patient is in a healthy patient, when they have 10 mm of induration, then we can say 90% confirm they are infected with tuberculosis. While with the induration of 15 to 20 mm, so we can say almost confirm they are surely infected by TB. While for those patients with below 10, which is 5 to 10, so maybe we can suspect the person is having TB infection, or maybe it's due to the BCG vaccine given previously. However, this tuberculin test produced several false positive and false negative results. For the diagnosis, normally we diagnose primarily using the sputum. Other than this, we can also use urine, CFS, and other part of diagnosis. This is because, as I told you, that this TB in fact is not only infect the lung, it may spread from the lung to other part of the tissue. That's why the diagnosis can also collect sample from the other part of the body, and we need 3 consecutive samples to confirm the infection. One of the way to detect this TB bacteria in laboratory is using this Gill-Nielsen staining. What we're going to do is, first of all, we apply this carbamazepine. After that, apply heating, followed by adding acid alcohol. From there, you can notice that this acid-fast bacilli, which is the Mycobacterium tuberculosis, they appear as pinkish red color. For the culture, the standard method to grow this TB bacteria is using this Lowenstein-Jansen media, or we call it as LJ media. This is the most commonly used. However, to grow this bacteria, it needs up to 8 weeks. We can also use molecular method to diagnose, or use PCR to diagnose this bacteria. For the vaccination, currently we are having this BCG vaccine to give protection for this TB infection.